Hello, everyone, and welcome to Affinity Proteomics and LCMS synergize Biomarker Discovery. I am Matt Thorne of LabRoots, and I'll be your host for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink. To learn more about our sponsor, please visit olink.com. We do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box in the bottom right and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues there if you're having any trouble seeing or hearing the presentation. I'd now like to welcome Valentina Meningin, Global Marketing Manager at Olink.
Thank you so much, Matt, and hello everyone, and welcome to today's Olink Multiomics Coffee Break, organized in collaboration with Discovery Life Sciences and LabRoots. I'm Vale Meneghin, Global Marketing Manager here at Olink, and I'm pleased to give you a quick overview on Olink Proteomics for those of you who are new to our company and technology. So Olink is a Swedish company by origin that in a few years has grown rapidly from a small Sweden-based company offering assay for a few hundred proteins to a global leader with a broad portfolio of flexible protein biomarker solutions and a library of high-quality, thoroughly validated assays that cover more than 5,000 proteins, with the ambitious goal of revealing the truth of human disease protein by protein. Our customers continue to drive significant advancements in the field with over 2,000 publications to date, covering a broad range of applications and disease areas. And as you may have heard, we are really proud that the UK Biobank Pharma Proteomics project chose the Olink Explore HT to measure over 5,400 proteins in 600 samples from the UK participants. And this collaboration between the UK Biobank and 14 leading biopharmaceutical companies represent the largest ongoing proteomics study. It aims to create the most comprehensive map of protein levels in the human body, significantly advancing our understanding of disease. Olink is now part of Thermo Fisher Scientific, the world leader in serving science, and this acquisition for sure underscores the profound impact of proteomics in advancing life science research, but also precision medicine. But we are not alone in this mission. We are doing this with our customers, the research community, and with a strong network of certified service providers. We are really proud to present to you the Olink Certified Service Provider network that is equipped to honor our commitment of delivering excellent data to our global research community. And we are really proud that Discovery Life Sciences is part of this prestigious network, having earned certification across the entire Olink portfolio. And this ensures that you can confidently outsource your projects to them, whether you are working on low or high throughput studies. And today, I'm super happy and delighted to welcome Dr. Jessica Moore, Senior Scientist at Discovery Life Sciences, who will present insightful data from her comparison study using both Olink Explore, Olink Explore HT, and LCMS on the same COVID-19 sample. So without further ado, Jessica, over to you.
Awesome. Well, thank you for the introduction. And today I'm really honored to share some of the work that we've been doing, combining Olink data and LCMS data. I'm going to start with just a brief overview of Discovery's capabilities to give you some background. And really the proximity extension assay technology, which might need very little introduction to this community, but for especially my mass spectrometry guests, the main figures of merit are that we have dual antibody recognition of a protein in its tertiary conformation, and that leads to really high specificity. And to those antibodies, we have a labeled oligonucleotide, and that tag can be PCR amplified, and that greatly enhances our sensitivity, especially for low abundant analytes and proteins. And as we mentioned, Discovery is a certified service provider for low, mid, and high-plex Olink assays. And that means we've demonstrated with our own data that we produce high-quality data that's concordant with the data that's generated by Olink at all plex levels. But also of note is that we have top-tier instrumentation for LC-MS analysis and enrichment as well. And that includes the Thermo Fisher Astral mass spectrometers and the Seer Proteograph SP100 automation instrument. And I share this just to say that when I show you LC-MS data that we are generating, it's quite good. And it's been generated on state-of-the-art technology. And this talk really focuses on the synergy between the two platforms. And that, by that I mean how each platform generates unique data that can be analyzed to understand a biological system. And each assay has many figures of merit, but as a high-level highlight today, mass spectrometry provides a direct measurement of peptides that are generated from proteins, and we don't have to have prior knowledge of these peptides. That allows us to capture a lot of information, including post-translational modifications or isoforms of those proteins. Now, Olink is an indirect measurement, which uses that antibody, but it has a PCR amplification step, which truly allows us to bring low abundance analytes up into the measurable range. So we wanted to really demonstrate these technologies, but we needed some sort of model system to test both platforms and compare the data. And we knew we wanted to work in a blood product because blood products are minimally invasive, they're biobanked, they're accessible specimens. A lot of people work with them. And also the plasma proteome is one that's famous for its analytical challenges, namely that our proteins span a very large dynamic range in this particular matrix, and that the top 20 proteins account for 99% of the protein content by weight. Albumins, transferrins, fibrinogens. Most of you probably are familiar with the problems, but we wanted to show how we can overcome the challenges, and we wanted a model system in plasma. So a lot of people ask us about immune response. It's a top request that we get because people are often interested in measuring cytokines in disease and clinical trials to measure adverse effects. It's a very popular way to study your biological system. And that's kind of how we ended on a small cohort of COVID samples. And SARS-CoV is a disease that unfortunately needs really little introduction to the community, but it's quite accessible to most people. Like, they understand the disease, they've had firsthand experience with the disease, and they've heard about implications of cytokines and the disease progression, even on the news, right? The cytokine storm. So COVID has become a really uniquely accessible biological system to study immune response because people are so familiar with it. And we're fortunate at Discovery to have a really extensive biobank. And so we were able to obtain and enroll a small cohort of samples. So we have 39 COVID negative patients, 39 COVID positive. Those negatives are self-reported as COVID negative donors, which later I will call undiagnosed. And they're from the Discovery Biobank. And here I'll note that this is not really intended to be a powered study of COVID pathogenesis, right? It's just providing example data to showcase the technology. So we did enroll COVID patients that exhibited severe disease in 3 loose categories, including those that were on ventilators or required respiratory assistance, which we called severe, those with a diagnosed bilateral pneumonia, and those with heart implications. And that included people who had heart attacks, tachycardia, cardiac arrhythmia, and that we called heart. And we've taken these samples through a number of Olink assays, including Explore-HT, Explore-3072, and the Target 48 cytokine assay. But for the first section of this talk, we are going to focus on the largest exploratory panel, which is Olink Explore-HT. So Olink Explore HT measures over 5,400 proteins from just 2 microliters of sample, and it requires 10 microliters of sample to go into the assay for dead volume. And that's really an amazing figure of merit when we talk about rare diseases or in cases where we just don't have a lot of plasma, or the samples are extremely precious. And so that's something major to highlight here. And we wanted to collect these data on these same COVID samples using LC-MS analysis as well. And to do this, we had to overcome that dynamic range challenge that is the plasma proteome. And we chose to do this using Seer's Proteograph product suite. Briefly, in this assay, biofluids are combined with a custom mix of functionalized nanoparticles. And these nanoparticles, because they're functionalized, are able to interact with the physical properties of proteins that are in their tertiary conformation, and they form something called a protein corona, a thin layer of protein on the outside of the nanoparticle, which is then digested into peptides. And this is all automated using an SP100 liquid handler. It's a Hamilton backbone, making the assay really rugged and reproducible. And so we were able to pair this enrichment with our ThermoAstral and Vanquish Neo LCs to generate our mass spectrometry data. But we're going to start our story today looking at that Olink Explore HT data. These particular plots were generated using the Olink Analyze Vignette, which is a package in R that's quite accessible. When we were first able to compare our data with a fixed limit of detection, which is generated from negative controls or blanks that are ran in our assay in the lot. And that basically tells us how many of the proteins that were measured were over the limit of detection. And from this, we were able to measure 4,193 proteins from our samples. So that's the number of proteins that we have confident measurements from that we can take into downstream analysis. We made a volcano plot comparing our COVID to undiagnosed populations, and we were able to find quite a number of differentially regulated proteins, 333 up, 683 down in this initial small comparison. And on the right here, I'm showing you some box plots of the top changing proteins from this data set, both up and down, where you're seeing the average MPX per group on the y-axis, and then the two groups, COVID or undiagnosed, in their own box plot. And if you're new to Olink data, whenever we talk about MPX or normalized protein expression, that unit on the y-axis, it's actually a log2 scale. So small differences in the number are actually quite large changes in the proteome. And so this was really great data for us to be able to show these big changes within these two populations. The LC-MS data was only collected on a subset, just the COVID samples. And so in downstream comparisons, we're actually just going to look at COVID to COVID. Using the Seer technology and the Astral, we ran a really quick 26-minute gradient on each nanoparticle. And worth mentioning, there are 2 fractions collected per sample. And we analyzed these data in the Proteograph Analysis Suite, which is a cloud-based platform that makes data analysis a little easier from these data. And we identified a total of 4,679 protein groups, and 1,909 of those are conserved within all COVID patients. So these data come from a total of 40,000 measured peptides. And they also represent 1,442 isoforms. And that's a huge strength of this particular technology, our ability to measure isoforms. On the right is an example of a waterfall plot, which basically just shows us where the proteins that we identified fall in relation to their theoretical abundance. And this shows us that we have really strong coverage of even low abundance proteins, so proteins that are expected to be in a low dynamic range. The bottom bar graph is showing us just the total number of protein groups that were identified per sample in the cohort. And so then what do we want to do? We want to put those data back together and see how they overlap. So in initial comparisons, we wanted to look at what we had identified with each technology and how they overlapped. And we saw that that was quite surprisingly low. So 13.5% of the proteins that were identified in both technologies. And to be included in this particular Venn diagram, the protein was detected in any of the samples in the COVID cohort. So we let as much in as we could. And this is maybe a bit surprising because these are the same plasma samples, but it is in alignment with what other researchers in our field have found. And it really supports that these are complementary technologies that are really accessing a different space within the proteome. So in this case, if we had used any one technology, we would measure 3,000, 3,500 proteins, but that limited number to having data on 7,700. And truly, when we consider deep proteomics for discovery, deep fishing experience, this is an incredible asset for biomarker discovery because we gain the ability to query twice the proteome by using a complementary technology. And I believe that this is really impressive data and a really important figure of merit, especially for our colleagues in biomarker discovery. However, we wanted to understand what these unique proteins were and what they bring to the study of a biological system. That's important if you're going to do two technologies. So the data analysis here could be quite extensive. It's a big data set. And what I can show you today is really just the surface of the data that we could mine. In our initial approach, we took these data into Web Gestalt to look at the pathways that were generated. And we can tell you that these did yield some pretty significant differences. They're quite different data sets. And to visualize it, we took it into Panther. And we made some string diagrams. So of note, the string diagrams that we're showing you today just represent the protein groups that were unique to each technology. So in the SEER, it's only things identified in SEER. The same with the Explore-HT. Proteograph is shown in teal, Explore-HT in an orange color. And what we can do is we can start to pick apart the pathways that are uniquely identified in each of these technologies. So here are some beautiful examples of pathways that were covered in both assays, but they brought unique partners to each of these known pathways. In purple, we have immune regulation between lymphoid and non-lymphoid cells. And in green, you see IL-4 and IL-13 signaling. So both of these are very much expected to be implicated in COVID samples, but we're showcasing here a unique overlap of the two assays within one known pathway to find different partners. And there is absolutely some complementarity between the two assays and some things that are completely unique to each technology. Here we have Explore-HT covering insulin receptor substrate mediated signaling. Didn't find that in the other pathway or in the other technology. And in SEER, some antiviral mechanisms and then SARS-CoV-2 known host interactions. And so these data are incredibly interesting because these are examples of pathways that we might have missed if we didn't mine our data deeply enough with our proteomics data. And although I don't have time to go very, very deeply into these data, the overarching message here is that even within shared pathways, each technology is bringing unique interactors to the pathway analysis. And it allows us to mine more deeply, and it allows us to strengthen our understanding of that biological system. And although these are all proteomics data sets, they're unique. I'm going to take a tiny minute to take a tangent to talk to you about quantitative analysis, because this is really a huge strength of Olink. And the Target 48 panels are able to provide both quantitative and relative NPX values from samples, and they only require one additional microliter of your biofluid, which is a great strength. Our team likes to test assays for ruggedness and reproducibility. Love doing that. And within this 80 sample cohort, we randomized both COVID and undiagnosed over 2 plates. This assay takes 40 samples at a time, so 2 plates to cover the cohort. And we analyzed it by 3 operators, which resulted in a total of 6 data sets. And here are these data presented at a very high level view, just to talk about detectability and reproducibility by operator. Here, orange samples are above the upper limit of quantitation. So they had too much protein to be within the linear measurable range. And the very dark purple is below the lower limit of quant. So we don't have a good data on those either, but the light purple samples are right within our measurable range. And so this tells us that overall, the detectability of this panel for these samples was very, very good. And we can look at these data with replicate measurements as something called a correlation plot. In a correlation plot, an ideal situation, our replicates give us the same data and they yield a line. Here, operator 1 is set as ground truth on the x-axis, and each replicate operator is set on the y-axis as purple and orange. And the R value is 1, showcasing the reproducibility of this particular assay. The 2 figures at the right are showing us in NPX space, the average signal of the 2 groups, and those are shown in orange, compared to the limit of detection in purple. Like to make these plots so that I know how far my signal is away from the limit of detection to help understand detectability compared to, the blanks. So finally, I have to show you another, albeit simple, volcano plot that takes these data that shows that cytokines are upregulated in COVID patients. Not groundbreaking, but definitely lovely to show that this technology works and works really well even on a quantitative assay. So to conclude this section, I want to highlight that although serum and plasma are absolutely analytically challenging, they really represent a rich matrix for proteomics. And analyzing plasma and serum with both Olink and mass spectrometry yields a truly unique data set that's complementary, and it deepens our understanding of the proteome in a biological system. We saw a 13.5% overlap, which was surprising. In this model system, we can take those samples, add just 1 more microliter of sample, and gain quantitative measurements of the cytokines with 1 microliter. And I think this really pushes the way that we as analytical chemists think about multiomics. Traditionally, when we talk about data stories, we're putting data together from complementary technologies, transcriptomics, genomics, metabolomics, but mass spectrometry and Olink, although both proteomics, are truly complementary technologies. And of note, this isn't a phenomenon that was unique to serum and plasma. We have seen it repeated again and again in alternative matrices as we put these data stories back together. Here in cerebral spinal fluid, which we commercially purchased, shared 14%. Summing those proteins all together, we covered 8,000 measured proteins. In urine, which was on Explore 3072, which is now kind of mid-plex, measures 3,000 proteins. There was a 14% overlap, a total of 6,000 protein measurements in this system from urine, a very complicated biomarker, non-invasive. And finally, from skin tapes, a total of a 14% overlap. I'm going to make this example bigger because it's really quite interesting data. So if you're not familiar, skin tapes are little stickers used in dermatology to sample the outermost layer of skin from areas of interest. And we queried a skin tape sample in multiple ways. We used the Protophy S-trap, which uses SDS, to interrogate an insoluble pellet or barrier proteins in the skin. Again, those are the proteins that allow your skin to be protective and do its job. And then with mass spectrometry and Olink HT, we looked at the soluble extract, the things that were soluble from the skin. And in total, we measured 8,600 proteins. 8,600 proteins from a completely non-invasive skin tape that just peels the outermost layer of the stratum corneum. And it does not require a biopsy from a patient. It doesn't hurt the patient at all. And these data are really impressive and incredible that we can generate these. We're going to take this to US HUPO next month. I'm happy to talk about them more, but Again and again, we keep finding the synergy of these technologies to deeply interrogate our biological systems. And truly, as we deeply interrogate our biological systems, we gain unprecedented information about the proteome, and we can use that information to further our understanding of human disease. So this is the amazing discovery team who work every day to use proteomics to tell biological stories. And the people on this slide were important to collecting all the data and enabling the science I showed you today. Also, we have to thank our Olink support team for the invitation and all their help along the way, and the Mass Spectrometry support team. And so with gratitude, thank you again for joining me on the talk today about how Affinity Proteomics and LCMS are truly complementary and can expand and deepen proteomics coverage. And with that, I'm happy to move on to any questions.
Thank you, Jessica, for your very informative presentation. Before we do get into those questions, we do have a couple poll questions we want for the audience to take a look at. The first one here that we have is, are you currently working on any protein biomarker studies? We'll give everybody a couple moments there to answer that first question. And while those answers are coming in, just a reminder that as we're going, we'll be doing that live Q&A here just in a second. So feel free to send in those questions that you have. Give everybody just another second or two on this first question. All right, those responses are slowing down just a second. All right. The next question is, if you are not, are you planning to start in the next 6 months? We'll give everybody just another minute or so to answer that. Okay. Next is, do you want to be contacted by Olink rep to learn more about the Olink PEA technology and how it can be integrated into your current omics workflows? Give everybody a moment there. It's another few seconds on that question. Do you want to be contacted by a Discovery Life Sciences rep to start your Olink project? Another few seconds on that question as well. And finally, which applications would you like the next webinar to focus on? And this is, you can select multiple options on this one. Let's give everybody a moment to answer that one. I'm going to give just a little extra time on this last question since you want to make sure you read all the options you have there. And I'll leave this question up as I just give everybody one reminder. So thank you everyone for answering those. We'll start the live Q&A portion. So if you do have a question, you'll see that Q&A box in the bottom right. We'll answer as many questions as we have time for, and you can submit them in that Ask a Question box. Anything we don't have time for today, our speakers will follow up with afterwards. So now I'd like to pass it over to Neil Skinner, a field application scientist with Olink.
Great. Thank you, Matt. And thank you, Jessica, for an awesome presentation. I love that. That was really interesting. So we got some good questions in the Q&A, so I'm just going to go through them here, and we'll see what we can talk about here. So first one is how is Target uniquely quantitative, and is Explore HT not quantitative? All Olink panels provide relative quantitation via NPX readout, which stands for normalized protein expression. So that's an arbitrary unit on a log2 scale. But additionally, some Olink panels, including the Target 48 platform, as well as some Plex and Focus assays, additionally provide absolute quantification in picograms per mil readout. So that would be the major difference there. Next, maybe this one's probably good for Jessica. So, could you explain why it is surprising that you had 13.5% overlap in proteins from the complementary technologies?
Yeah, I mean, whenever you go with the same sample through two things that are going to measure proteins, like, I don't know, I would have thought that I would get the same answer, right? Or close to. I think one of the beautiful bits about Olink is what you're going to get, right? Like, the 5,400 proteins that are going to be queried, and you'll get a measurement from them. Mass spectrometry is always surprising. But I mean, maybe I can see where you could say maybe you shouldn't be surprised. They're very different. The places that they mine are quite different. But it was just something that I think, especially as an analytical chemist, I just thought it would be higher. And I didn't realize going into this how complementary they were going to be, but truly, again and again, they are.
Thank you. Next question. Could you provide reference information as to how your skin tape work was done, most notably the extraction of proteins from the tapes for the Olink and LC-MS methods?
Yeah, that's a hard question. With blood, sweat, and tears and a lot of R&D, we worked with a partner, and we're scraping them with a cell scraper to remove the tape, the skin pellets from the tape, and that's how we're generating pretty good skin pellets. And then again, this is my poster at HUPO next month. Happy to talk through all the nitty-gritty details of how we're doing that, but yeah, we physically remove the skin with a cell scraper.
Right. Next question. Did you compare the quantifications for Olink and MS for the 1,051 proteins that were measured by both platforms? And if so, what did that correlation look like, on values and such?
I didn't do that, but I will now. It's an excellent suggestion. Yeah.
Next question. I think I can field this one, but definitely let me know if I get it wrong here. So how is the LOD defined for Explore HT? So in this case, I believe I heard that we used fixed LOD. Is that correct?
Yep.
So very briefly, there's two major ways that we offer to determine LOD for Explore HT projects. One is a project-based LOD, which if you have a sufficient number of negative controls, then you can use a function to calculate that way. The other option is a fixed LOD, on our website from validation and QA studies. So that's another option. It's probably recommended if you don't have a particularly large study that you're looking at. Let's see. Next question. Especially regarding the proteomics from the skin tapes, can you detect bacterial, I think it was proteins was supposed to be there, from the skin microbiome, which could explain the differences?
So definitely did not search against any, we just searched against human, right? So if they were, the peptides might have been there and we might have not seen them because we didn't search for them, right? So that's another great suggestion. Like, when you go into deep proteomics data, you're making these informed decisions, and we just searched against humans. So there could be a lot more there that we don't even know about yet.
Next question. Let's see. Do you have publications that have done a comparative analysis of Olink and LCMS, like in the COVID example?
Yeah. So they're coming out, right? We definitely want to publish these. We haven't quite yet, but the Kuhn Lab just put out a preprint that did some of this. And so that is searchable and available. That's probably the best reference I can give you at this time. But I'd expect more to come in the year as more researchers get their hands around these two technologies.
Yep, some good questions. Thanks, everybody. Let's see. Next one. Let's see. Did you mix the plasma sample before you withdrew aliquots for the Olink and MS assays?
Yeah. So we had aliquot, so when at the time of the blood draw, when the plasma was double spun, it was aliquoted. So we used different aliquots mixed. I think that, I don't know. That's how the samples were processed. So yeah.
Let's see. Next. Okay. Either one of us could take this one. Let's see. I'll let you take a stab at it if you like. I'm happy to back you up. Can you briefly describe the QC steps of proteomic data? I have been working with Olink HT data, and I'm using Z-score transformation for data normalization. I am not observing such high difference in log fold change as seen in your volcano plot for the COVID project.
Yeah. So the first thing that I will say is that when we designed this study, we designed it to be a tour de force of significant differences, right? Like we took people that were in the intensive care unit with COVID and compared them to normal Alabamians. Like, Lord, if I didn't get high fold changes, like when would you? Right. So whenever we look at other biological systems, it definitely is more nuanced, and there's some decisions that you may have to make. And I think that in the case of whenever we've looked at HT data, there are sometimes that we do different normalizations depending on what we're seeing in the proteome. Yeah, and so I think that there's lots of ways that you can process those data downstream to help you whenever you make informed decisions about, like, you can plot like average MPX, and if you see a difference in your two populations, bring it back down, which might be what you alluded to that you're doing. Of note, if you've not done this yet, Olink has a tool. It's a web-based tool called Olink Insight. And it has a power calculator. And so a lot of times we'll take things into that power calculator and figure out if our studies are powered. And alternatively, you can also look at the normal ranges that are provided there to see how your data aligns with the UK Biobank, which, my goodness, what a resource to be able to query as researchers, especially if your cohort's on the small end. So those are my suggestions for looking at those data.
Yep. It just, the only thing I would add is just I wouldn't be qualified to talk about QC of mass spec, but just very briefly, QC for Olink data is looking at a combination of internal synthetic control performance in both the experimental samples as well as external control samples that we provide. And that rationale is listed in the manuals for our software online as well. But that's essentially how the QC works for the PEA data from Olink. Let's see. Next question. You mentioned isoforms were detected greater than 1400 by mass spec, and I wonder what was the identification strategy?
Yeah, so we're searching against the Swiss Prot reviewed human database. And so those all, and it's again, mass spectrometry, you're fishing against the database that you're searching against. And so the better our databases, the better our results. But that's absolutely true. Like maybe there's a rare isoform that is not in canonical human proteome. I would miss it, right? Because I'm not searching against it. And so all we know is the ones that we're in Swiss Prot reviewed human proteome.
Is there any other machine that we can use to measure the same data as CMS?
I think there are lots of ancillary technologies in proteomics that, we talk about. And these are always tricky decisions, right? Where you say, what analytical platform am I going to use to perform my study? So that's kind of a non-answer, but I think there's a lot of stuff out there. And I'm sure that they all will bring unique data to these data stories. And, these are the two that we have used and we've had a lot of luck with. And I'm sure there are others that are out there as well.
Let's see. What was the major difference between the Explore HT seer proteograph and traditional chromatography?
Tricky question. So the major difference, I think, if I were to tell you like why I think they're acting differently, is that with Olink we're able to PCR amplify, and I'm a mass spectrometrist. I maybe didn't lead with that. That's my training. And so I didn't appreciate the ability of PCR to bring things that were not measurable because they were very low copy up into the measurable range. And that's been, I'm a believer now, right? I went in maybe a skeptic, I believe now, because it is a well-designed assay, right? Like having that PCR implication really, really helps. And of course, mass spectrometry, you don't have to know the question that you're asking. You can tell your sample, tell me your stories, and then we can put those stories back together again.
Okay. Let's see. Have you looked at post-translational modifications from the seer data?
We did not search for PTMs. So those are probably in there. More data to mine. Yeah.
Next question. Did you check the data distribution, such as coefficient of variation for Olink and seer data?
We have before, right? Where we have replicate measurements. Big data gets tricky, right? Because you have to run the assay a multitude of times. We've done it for target, right? Like the target data that we have and the CVs look mostly good. Analytes vary, right? Most of them are quite low CVs. There are some that are a little more low abundance, a little larger CVs. And we've done it on some of our mass spectrometry data as well whenever we collect replicates. That wasn't the case for these particular data, but we have them for other systems.
Let's see. How did you fractionate the insoluble and soluble proteins in the skin tape samples?
Yeah, we didn't, right? We just used those two different vendors, direct digestion solutions, which are both beautiful, Protophy S-Trap and then Preomics IST kit. They're both kits, right? Really easy, really accessible to use. And then they get those data from no enrichment, which is mayhaps worth mentioning as well.
And then there is a question about what was the reference for the Kuhn lab? There's some uncertainty if the last name was spelled correctly, Coon. Yeah.
We'll find it and email it. It's a preprint, so I think it's on bioarchive. But let's answer that one offline and send it.
Very good. Let's see. A technical mass spec question. So was the MS acquisition in DIA or DDA mode?
DIA. And so that is how the Astral runs really well. It is lightning fast and can collect DIA data quite quickly.
And then it's sort of an admin question. Will you be able to provide the text of this presentation and the QA session for us? I don't think the QA session we would be able to provide in text, since it's all answers have just been verbal here. But I presume the slides will be shared, perhaps?
For sure, we will share the recording. Yes, Neil. Not the
Recording.
slides.
Oh, okay.
Yes.
There you go. The best video. Okay. And let's see. Okay, that's a good kind of general question here that yeah, Jessica, I'd be happy to hear your perspective on this. Is yeah, how do you design studies to ensure the quality of the biomarkers?
Yeah. So I think that especially all of us maybe know this, right? Like all of our friends here in biomarker discovery, that these are really screens, right? And when we do screens, we get candidates and we must validate those candidates. And so all we can do is make sure that we've designed the best study that we can. That power calculator, anything that you can do to make sure that you have high quality samples, you know, the outcome of your samples. Like anytime we're doing deep proteomics and we're fishing, we want to set ourselves up for success with the highest quality samples going in, rigidly quality controlled assays, so that at the end, when we get these candidates and we go to validate them, we have confidence in them. And I think that both these assays, and especially with how well controlled Olink assays are with their internal controls, external controls, it's really lovely data. You have a lot of confidence in the candidates that you take forward to those, you know, more expensive steps of validation.
Great. And then last thing here. So this is not quite exactly a question, but a comment. This will be the last thing we have time for. Everything else we'll try to reply to via email. But fabulous job, Jessica. You are a natural behind the camera. Flawless presentation.
Hey, thank you.
Like to hear that one.
Right. Well, thank you very much to everyone, to our speakers and everyone else for their time and the important research. We would like to thank LabRoots and our sponsor, Olink, for underwriting today's webcast. Before we go, I'd like to thank the audience for joining us and for all those questions. Any questions we did not have time for today that get submitted during the on-demand period will be addressed and via the contact information you provided when you registered. This webcast can be viewed on demand. LabRoots will alert you via email when it's ready for replay. We do encourage you to share that email with your colleagues who might have missed today's live event. Until next time, thank you very much and goodbye.